Hello to everyone who's watching this. My name is Rohit Kapila. I am a final year PhD student at Dr. Prasad's group at Indian Institute of Science Education and Research, Mohali. Today I'll be talking with you about one of the projects that I did during my PhD, the aim of which was to look at the evolutionary consequence of adaptation to poor developmental environment on adult stress response. I'll start by explaining the word stress, since it can mean different things to different people, especially after the year 2020. So, for the sake of this talk, I'll be sticking to the definition of stress that was used by Hoffman in 1991, which states that stress is any environmental factor that can cause a potentially injurious change in the organism. Now, the fitness of an organism depends on its ability to cope up with the stress that it encounters during its life and still reproduce to the maximum of its capacity. Now, studies have shown that a lot of bodily resources goes just into maintaining general body condition during the time of stress. Therefore, the general expectation from individuals that are developing in a poor developmental environment is that they would be bad at handling environmental stress, majorly because of the poor developmental resources. However, our understanding of evolutionary consequence of adaptation to poor developmental environment is not very great. So the aim of this study was to look at what happens to the adult stress tolerance ability when populations are grown in poor developmental environment for a single generation versus when populations are adapted to poor developmental environment for several generations. For that, we decided to look at heat stress tolerance ability, and we have worked on model of Drosophila melanogaster, which is a holometabolous insect. Its life cycle is divided into various larval stages. Larval stages are the primary feeding stages for them. There are three larval stages, then there's one pupal stage, and that is followed by the adult stages. The populations of Drosophila melanogaster on which we have worked are called MBs and MCUs. We have four replicate populations of MBs, that is melanogaster baseline population, MB1, MB2, MB3, and MB4. And some 250 generations ago, from each MB1, MCU population was derived. MCU stands for melanogaster crowded as larvae and uncrowded as adults, which is basically their maintenance regime. MBs every generation, they are grown in very healthy larval environment with 60 larvae in 6 ml of food. So there is enough food for each larvae. Whereas MCUs every generation are selected for adaptation to poor developmental environment. How do we induce poor developmental condition in them? Well, we put 800 larvae in 1.5 ml of food. So basically there is very little food for every larvae to have. And then somewhere one and a half day into the larval culture, food completely runs out and the vial is empty without food. As a result of which, only 70 to 80 larvae of these 800 makes it to the adult stages, and those 70 to 80 larvae they reproduce to make the next stage. In the adult stages, both the populations, MCUs and MBs, are maintained in exactly the same way, with an adult population size of 2400, and MCUs therefore have been selected for adaptation to poor developmental environment for more than 250 generations now. Whereas MBs every generation, they are for last 250 generation, they are being held in a very good developmental environment. So in order to look at their adult stress response, we use the following four treatments. Where in two of those four treatments, MBs were grown in their own developmental densities, that is MB in low density, 60x in 6 to 8 ml of food, whereas MCUs grown in high density, 600 individual larvae in 2 ml of food. We also had two more treatments where MBs were maintained at high density, that is 600 larvae in 2 ml of food, and MCUs at low density, where 60 eggs were there in 6 to 8 ml of food. So this would give you, when you look at MBLD, MB low density, you would have an idea about the general response of the population. When you will compare MBLD with MBHD, you would get an idea of what a single generation of crowding event, larval crowding, does poor developmental environment does to the adult responses. When you will compare MBs from HD treatment to MCU from HD treatment, you would get an idea of what adaptation to uh, poor developmental environment for 600 generation for 250 generation does to the adult stress response. And when you will compare MCU HD with MCU LD, you will get an idea of what how plastic the evolutionary response is. So with these four treatments, we went ahead and did the experiment. Experimental procedure was pretty much straightforward. Eggs were collected from for all the four treatments in such a way that on the day of experiment, all the adults were of same age. Now the population were, well, all flies from all the four treatments were subjected in separate assay to different heat shock treatment. In one of the treatment, they were subjected to 37 degrees Celsius for an hour, and in other treatment, they were subjected to a 38 degrees Celsius temperature for 1 hour. It is very important for you to note here that these populations are maintained at a temperature of 25 degrees Celsius, and in their evolutionary history, they have never seen anything beyond 25 degrees Celsius. In fact, the literature suggests that in Drosophila, anything above 35 degrees Celsius is considered as heat shock. At 39 degrees Celsius, when we subjected these populations for 1 hour, we found out that all of them died. So 37 and 38 degrees Celsius gave us a nice window to look at the difference in the mortality. Therefore, we use these two temperature treatments. So the heat shock was given at 37 degrees Celsius and 38 degrees Celsius for 1 hour. And after that, flies were again kept at 25 degrees Celsius for recovery. And post recovery phase of 24 hours, we counted the number of dead individuals in all the treatments. The population, well, the sample size was 100 individuals per population per treatment per sex. The results were following. As you can see in the left panel on your screen, there was an effect of selection history on heat stress tolerance ability at 37 degrees Celsius. As you can see on x-axis, there are the two population, MCUs and MBs, and on y-axis, you can see the proportion survivorship. Clearly, the proportion survivorship of MCUs, the populations that are adapted to poor developmental environment, MCUs, it was better than MBs at 37 degrees Celsius. When the assay was done at 38 degrees Celsius, as you can see in the right side panel, you would realize that MCUs continued to have a significantly better heat stress tolerance ability at 38 degrees Celsius. So adaptation to poor developmental environment led to evolution of improved heat stress tolerance in the adult stages. Surprisingly, there was no effect of density treatment. As you can see in the left panel, on x-axis, HD stands for high density and LD stands for low density. There was no significant effect of density on proportion survivorship. Similarly, at 38 degrees Celsius, there was no significant effect of density treatment on proportion survivorship. When we looked at the interaction of density cross selection history, we found out that the MCU high densities had a significantly better proportion survivorship than rest of the treatment. As you can see in both left and right side panels, the black bars here are for MCU populations, the populations that are adapted to poor developmental environment, that goes through poor developmental environment every generation. They have a significantly better survivorship as compared to other three treatments, MCU HD, at both 37 degrees Celsius and at 38 degrees Celsius. The tallest bar represent MCU HD. When we dissected this result by sexes, we realized that the major effect is differences because of the male survivorship. As you can see in the left panel, there was not much difference, well, no significant difference of proportion survivorship um, in both the population in any of the treatments. Whereas in the right side panel, you realize that males of MCUHD, that the black bar from the HD treatment is tallest, signifying that MCUHD had the better survivorship than rest of the three treatments. And same was the result at 38 degrees Celsius. As you can see, there was no significant difference in the female treatments, but in male treatments, MCUHD, the black tallest bar, shows the most significant proportion survivorship improvement as compared to the rest of the treatments. So what do we know from here? We know that adaptation to poor developmental environment, when organism goes through poor developmental environment every generation, the males of that population end up having significantly better heat stress tolerance ability in the adult stages. It is again very important for you to here note that these population in their ancestral history, they have never seen anything beyond 35 degrees Celsius. So there was no selection happening for increased tolerance of heat shock in the adult stages. Yet there is a correlated evolution of adult heat stress response in these population. So we thought why this would be the case. When we looked at the literature, we found out that in Drosophila, larval crowding is perceived as stress. and it induces the expression of stress response genes, which made us to think that if the genetic architecture of stress response gene is not remodeled across the different stages of life cycle, then probably the expression of HSP7, expression of stress response genes in the larval stages is carried forward in the adult stages as well, which gives improved adult survivorship against stresses in the selected population that are selected for adaptation to larval crowding environment. So we went ahead and we looked at the HSP70 expression. What is HSP70? HSP70 is one of the very famous heat shock protein. It is a chaperone protein. What it does is whenever there is any kind of stress, the misfolding of proteins happen or unfolding of protein happens, then HSP70 protein, it's a chaperone protein. It helps the unfolded or misfolded proteins to refold properly and thus enabling the chances of population to individual to survive better. So we thought that probably because it's heat shock induces misfolding of protein, increased expression of HSP70 must be happening in the adults of our selected population. So we looked at the HSP70 expression using the same experimental protocol, but this time the temperature at which the heat shock was given was at 35 degrees Celsius. This temperature does not induce mortality, but induce the expression of HSP70 in these populations. Surprisingly, we did not found any effect of selection regime, density treatment, or their interaction on HSP70 expression. As you can see in the left panel for females and right panel for males, there was no significant effect of any of these factors. So we thought that probably there is something happening in the larval stages. So we grew the larvae of these populations in high and low density treatment to look at their HSP70 expression, and we found out that there is a significant effect of selection history on HSP70 expression in the larval stages. When the larvae were crowded and uncrowded, MCU population, as you can see on the y-axis, it represents delta CT values. Lower the delta CT value means more the expression. So population that was selected for adaptation to poor developmental environment or crowded larval environment, they were expressing more HSP70 in the larval stages as compared to the populations that were coming from a healthy developmental environment. And so was the case when we looked at the selection cross density interaction, that in both low density and high density HT treatment. The black bar, that is for the population that is adapted to poor developmental environment, they were expressing more HSP70 in the larval stages. So probably as a result of adaptation to, well, going through a poor developmental environment every generation, these populations are selected for increased expression of heat shock protein or other stress response proteins that we have not looked at. As a result of which, there is a carry forward effect of this increased expression in the adult stages, which has improved the population's stress tolerance ability in the adult stages. Um, well, the takeaway message from this experiment was that the general expectation that people had up till now, that populations coming from poor developmental environment would be bad at handling environmental stress due to lack of resources, is not true at all when you look at this from an evolutionary perspective. These experiments prove that stress response can evolve to improve in resource limited conditions as well. Thank you for your time. I would like to say thanks to all of the people of Evolutionary Biology Lab, especially to these three people, Mayank, Podar, and Shreya, who have helped me to run these experiments. And special thanks to all the people from different labs of ISER Mohali who have helped me in running this experiment. Here are my contact details if you want to contact me for any further questions. Thank you.